Good morning, everyone. It is with a great pleasure to introduce Katrin Amunts. Katrin is a very distinguished and internationally recognized neuroanatomist. A brief glance to her CV is sufficient to appreciate that not only is she phenomenally productive, but also most of her papers are published in the top research journals. Over the years, she has made many significant contributions to the field. Katrin Amunts is a full professor for brain research and director of the Cecil and Oscar Both Institute of Brain Research at Düsseldorf University since 2013, as well as director of the Institute of Neuroscience and Medicine in Jülich Research Center. Since 2016, she is the scientific research director of the Human Brain Project, and she did a postdoctoral fellowship at the Cecil and Oscar Both Institute of Brain Research at Düsseldorf University. In 2004, she became professor for structural functional brain mapping, and in 2008, a full professor of the Department of Psychiatry, Psychotherapy, and Psychosomatic at the Aachen University, as well as the director of the Institute of Neuroscience and Medicine at the Jülich Research Center. The variety and excellence of her numerous contributions are reflected in the frequent citation of her work in the scientific literature. In particular, her studies focusing on the organizational principles of the human brain represent a major advance towards understanding the human brain. As an example, Katrin and her team has created the cytoarchitectonic Jülich Brain Atlas. And this is important because one of the major questions in neuroscience is how neural circuits contribute to the functional organization of the brain, giving rise to cognition, and behavior. So one of the first steps is to define its detailed structural design and to map its connection matrix. And to this end, maps of the microstructural segregation of the brain are key to understand the biological substrates of brain function and diffusion. So, Katrin, I'm here. So, to this end, the Brodmann's cytoarchitectonic map that was published in 1909 was one of the first maps and is still widely used. And this is a problem because this map has several important limitations. For example, it consists of a single brain map of a left hemisphere. Inter-subject variability is not reflected in this map. Modern maps of the brain indicate that the number of cortical areas is in the range of 180 or more compared to the 43 areas of Brodmann's map, and subcortical structures are not part of the Brodmann's art. So it was very important to create a modern map of the brain, and that is what Katrin and her team has created an atlas based on serial historical sections of the brain that provide some critical elements which are very, very important for better understanding the brain. One is map the areas of 3 dimensions, integrate recent knowledge about brain parcellation, consider variation between individual brains, which this is also critical because the variability, and also is web-based links to other resources and database. So this is a major contribution, this Jülich Brain Atlas, and that was published in Science last year. So I would like now to go ahead and give the floor to Katrin. Katrin, thank you very much.
Thank you very much, Javier. So I'm really impressed and by this very kind introduction. And indeed, I would like now to give you an overview of my project, the Human Brain Atlas, and would like to ask for the first slide. Okay. So what does it mean to build an atlas of the human brain? And we can think, of course, about something like this, but we could also think about something like Google Earth, that is perhaps the modern version of a digital atlas. But I think when we started our Atlas projects now more than 25 years ago, we were very much inspired by the old and early cartographers. And one of them is shown here. This is Alexander von Humboldt. And he was a polymath, naturalist, geographer, explorer, and he emphasized the unity of nature. And he also introduced quantitative measures. So I think what he did for geography such a long time ago is also now very valid and has a high relevance for modern brain research. And in particular, I would like to emphasize that he has created the first multimodal atlas of the world. And when he went to South America, he did not only draw maps of the geographical characteristics, but he also measured the height of the mountains. He analyzed the plants that are there, and he did first analysis of the climate and of the temperature. And he brought everything together in a very comprehensive way and created this type of atlas, which I think is really exemplary. And a little bit later, starting with the 90s of the 19th century and then the beginning of the 20th century, there was a couple of researchers in Berlin who started in a private apartment, in a lab that was in a private apartment. This is Oskar and Cecil Vogt. And at the window, you can see Corbinian Brodmann, about which Javier just talked a minute ago. And these researchers, in my view, really revolutionized brain research and brought it to a modern basis. Why do I think so? Well, their concept was, from the very beginning, an interdisciplinary one. And when the Vogts created a brain research institute in Berlin, they had different departments which are listed on the right side, including anatomy and architectonics, histology, but also electrophysiology and neurochemistry and pharmacology. And what I find extremely exciting, at the beginning of the century, there was experimental genetics. This was long, long before the DNA was identified. There was physiology, psychology, phonetics, biophysics, and there was a department for photography and documentation, as well as links to the hospitals at the site and clinical examination. So Vogt was convinced that it is only possible to come to a deeper understanding of the human brain when we bring together the different disciplines, and he did it under the roof of this early brain research institute. So in that respect, I would feel a little bit like a successor of these early approaches, and Photoshop makes it possible to bring me into a very lively discussion with Cecile and Oskar Vogt. So I would see that we are now continuing on this way, and that we are introducing new methods and new approaches that significantly improve the way how we can map the brain and how we can analyze the brain. And one is intersubject variability, also mentioned by Javier DeFelipe. And when I did my first mapping project of the visual cortex areas V1 and V2, this is a primary and secondary visual cortex, and mapped them in the context of their brains, then we found that there's a considerable variability in the size and in the extent of these areas when we compare different postmortem brains with each other. And of course, the question occurs, what has this variability to do with function? Does it have anything to do with function? And what precisely is the relationship to it? So in order to address this intersubject variability and not only to exclude variability as a factor of noise from experiments and to make the cohorts more homogeneous, we developed so-called probabilistic cytoarchitectonic maps. And they show for each voxel, for each position in a certain reference space, what is the probability that a certain area can be found precisely at this position. And red means, for example, that we found this particular area here in all brains that we have studied. And all brains means here always 10 brains contributed to the map of one area in this atlas. And on the right-hand side, you see a so-called maximum probability map where each position of a reference brain has been assigned to that area that has the highest probability to be there. Unfortunately, this is an extremely time-consuming process. So mapping is very slowly, and although we are developing tools, it's not so much faster than 20 years ago. For one area, we need about one person year. And meanwhile, we have mapped about 170 areas that are in progress or have been already published. And we decided from the very beginning that we would like to share these areas with the community and to provide it as a resource tool also for others to address, for example, brain function as we've seen it in functional imaging studies. And the Human Brain Atlas that has been developed in the context of the HPP, this allowed to scale it up and to develop tools that really make it easy to access these data. And one of the latest developments, these are so-called surface-based maps, which you can see here. And these surface-based maps are very frequently now used in the functional neuroimaging community to localize findings of functional activations that we can see in cognitive tasks. The difference to other maps is that we do not just have the areas visible there, but as you can see on the left-hand side, there is many more information behind each area, like the receptor architecture, illustrating how the molecular organization is, or the connectional structure, functional connectivity, which relates to what Carl Friston has said in his last talk. So the idea was to build a multimodal atlas framework where we integrate information coming from different labs in Europe and worldwide, and also reflecting different aspects of brain organization from the molecule through cellular networks up to large cognitive systems. So that was the idea. And of course, there is a huge effort behind that made it possible to create tools for visualization, for data annotation, for data processing, to make it findable and reusable. And we call it fair data. And this is, again, one of the, in my view, very important steps that the HPP did. So bringing these data together allows at the end to compare the Jülich Brain Cytoarchitectonic Atlas with other kinds of information, for example, on the connectivity of brain areas, such as Jeff Marjan and Cyril Popong are doing it at Neurospin, or with functional segregation of the human brain, such as Bertrand Thirion and his team are doing it. And we hope that by bringing these different aspects together, we come to a more comprehensive view of how the brain is organized and what does this organization mean for function. And this is also important to better understand dysfunction. And therefore, we are applying this atlas in order to better localize findings that we see in brains of patient data. And this coming from a tumor surgery at Düsseldorf hospital. And here we have compared the areas with stimulation sites of patients undergoing surgery. And we would like to further inform surgeons about the structure of the areas that they touch when they stimulate, but of course also about the functional relevance of the areas through the atlas. We would like to go even one step further, and this is particularly important when we think about modeling and simulation, for example. Then it's not enough just to visualize the brain, to rotate it or to slice it, but then we need a programmatic access. And based on Python scripts, we have now built a possibility that researchers can extract data. And you need only a very few lines in Python code, for example, in order to extract a whole bunch of receptor data for a certain architectonic area. And this data automatically can go into modeling and simulation, for example. And it also helps us to better analyze data from living human subjects, from patients, for example, that have undergone positron emission tomography. And here we have the possibility to observe the molecular status of the living brain when we are doing challenge tasks, for instance, or when we ask people to solve some cognitive task. And by using this Zebra tool, we could connect the cytoarchitectonic maps with the underlying receptor architecture as obtained from receptor autoradiograms of more than 15 receptor types of many different receptor systems, and map this, as you can see here, to the brain of patients that have been analyzed through PETs. And we have, by this combination, created a situation where we can better understand what is, for example, the mechanism of a certain drug acting to the brain, and we can better distinguish with respect to which receptor and with respect to which neurotransmitter this drug is acting. And this was rather quickly done using these new tools of analysis. And I think this is really a step forward as compared to other brain atlases that we see in the world. And I would like to invite you to learn more about this way of analyzing PET data in our breakout session on atlasing 12:30 with Roshan Kuhls and Timo Dickscheid and many other colleagues who give you an introduction and help you to better understand what atlasing tools today can now do beyond just visualization of interesting aspects. We would like together to go, however, to higher spatial scales. And why do I think that this is necessary? Well, because when we want to understand the cellular mechanisms and the molecular mechanisms and even the synaptic, as Carl mentioned, then the spatial resolution is important, and we need to link such information to the localization where it is in the brain. And this is really a problem to address a complex system and crossing many, many spatial scales. If you think about that synapses are in the angstrom and nanometer scale, neurons are in the few micrometer scale, but the brain is many centimeters long. So to bridge these different scales really needs a lot of compute power, but also conceptual work. And what we did here is we tried to reconstruct in 3D at the histological level of 1.5 micrometer in plane resolution, the hippocampus. And the hippocampus, as you know, is a very important hub for learning and memory, and in particular in neurodegenerative diseases like Alzheimer, it suffers rather early and patients lose their memory. And what we have done here is we have reconstructed the human hippocampus of the temporal lobe in several hundreds of sections with extremely high spatial accuracy to build in this information into the so-called big brain template, which is a high-resolution anatomical template of the human brain. And the different colors that you see here, they are not just indicating so to say, different fibers, but they code the direction of fibers. So polarized light imaging in 3D allows you to identify the direction of fibers in all three directions. Plus, it gives insight to the cellular architecture, as you can see here when we go to very high resolution. And we are now moving in the temporal lobe through the angular bundle, which is a very important fiber pathway, to the subicular complex, and you see that the fiber architecture changes quite significantly, and you get an impression of the richness and also of the complexity of fiber orientation that we see in the human brain, and that goes, of course, by 1-2 orders of magnitude beyond that what we can see in the living human brain, and helps to get better insights into the connectional structure of the human brain. And this is so important because we cannot only infer from connectivity studies that we are doing in rodents or in monkeys to the human brain, we also have to address the human brain, which is not just a scaled up animal brain. It has its own principles and concrete aspects of connectivity. So this is what we like to address here. And this, of course, gives us very new insights into brain organization, but it helps us also to develop new tools that can be then transferred to the clinical context. And what you see here, this is a MR image of a patient that has been scanned. And you can get an understanding that the contrast is not always so great. It's not so easy to distinguish all these smaller subnuclei and nuclei in the human brain. And it's also not easy, and for most of the cases also not possible to identify brain areas. So we use our maps in order to identify brain regions to a more precise extent. And we are developing new methods to better image the living human brain. And this is done here by the colleagues from CEA, who developed diffusion mappings that are very precise, that go down to the millimeter scale, and that really give us an impression of where the fibers are going on. But here in a particular patient brain or in a healthy human brain. And we can then apply the same type of imaging studies to do also postmortem studies. And of course, the spatial resolution goes up, and we can better understand and inform in vivo imaging by postmortem imaging where the fibers are being located, what their directions are, where they are crossing, and where they perhaps are only kissing, touching each other, and do not cross. And these are very hard problems when the connectivity matrix of the human brain is being modeled. And by having this multi-scale approach from the living human brain to postmortem whole brain mapping to PLI imaging, we think that we have very powerful toolkit now created that allows us to go really into the details of connectivity. And this is particularly important when we think about regions like in the mesencephalon, where we have many, many cells that are distributed, where the contrast is rather low, and when the nuclei are located very closely to each other. So even in histological sections, it's not so easy to distinguish these nuclei. And you see on the left-hand side an anatomical illustration of polarized light imaging from the red nucleus and the substantia nigra and the subthalamic nucleus. And you all know, of course, that the subthalamic nucleus is a frequently used target for deep brain stimulation. And surgeons want to place the electrode as precisely as possible into this region, which is a challenge. And we hope in the future that by providing this more detailed information at the level of connectivity and at level of the cellular architecture, as shown on the left-hand side, where we have mapped each single nucleus of one cell in the subthalamus, so to inform models that we can use for neurosurgery to improve the outcome of surgery. And that means we have to reconstruct volumes of interest at the 1 micrometer spatial resolution, because this is a resolution where we can see cell bodies. And we did it here, as you can see it here, for one region of the visual cortex, where we have aligned at the cellular level several hundreds of sections. And the advantage that we have is huge. So when we have a look to the big brain template with a spatial resolution of 20 micrometers in every plane, then you can see here that we identify the cortex, the layers, of course, and we have an idea of how the cells are being packed, where are more densely and less densely packed regions. But when we increase this to 1 micrometer resolution, then we see individual cell bodies, and it's a completely different world. It's like having a new telescope and looking to a different galaxy. So along the same lines, we would like to improve microscopy and make it three-dimensional. And therefore, we have developed a technology and workflows that allow us to image single histological sections with their whole depth, which is 20 micrometer. And then, so to say, we have a stack of many, many thousand of such histological sections, and have to identify cell bodies that have been cut into 2 pieces during histological processing. We have to identify them, we have to match them, and then in the last step, we have to align these different cells throughout a whole number of histological sections. And there can be 6,000 to 7,000. So saying that, I would like to stress the point that we really have a gigantic optimization problem that we would like to solve. And when we have in mind that there are 86 billion nerve cells in the whole human brain, and they are distributed through the brain in a very systematic way, then we have somehow to identify cell bodies and align them in order to come to such high resolution images. And this requires, of course, high performance computing with respect to the storage space, with respect to the memory that we are addressing, but also with respect to the compute power. And this is possible again through the human brain project platform and the Phoenix infrastructure. So at the end, we would like to integrate this type of data, and for surgery, deep brain stimulation, we would like to bring in anatomical information through probabilistic maps, PLI information, cellular information, and then map this to patient brains undergoing deep brain stimulation. So with that perspective, that is not yet here, but I'm very optimistic that we will come to this new era of brain research and also application of human brain research to medicine. I would like to thank my collaborators in my teams in Düsseldorf and Jülich, and the many collaborators that I have in the Human Brain Project. And I would like to thank you for your attention. So I would not only like to thank you for your attention, but also perhaps to ask you for questions and for comments. And I'm receiving it here through the iPad. And the question is, you mentioned that the probabilistic maps were made using 10 brains. What is the variation between these brains in terms of female, male, young, old, genetic background? How likely is that a random new brain would indeed fit into this data? Well, the last question we have experimentally verified. So we have exchanged one of the 10 brains and took an 11th brain or a 12th brain. And at the level of probabilistic maps, there are changes which are below the millimeter range. So the probabilistic maps are quite robust in reflecting the intersubject variability that we see in these brains. We see variations between male and female brains, and in our paper last year in Science, we have illustrated this. There are variations between the two sexes, and we really only do have information about male or female brain, no further information. With respect to variability in a broader sense, well, we see that variability is very different in dependence on where you are in the brain and to which level are you looking. So primary areas seem to be less variable. Higher associative areas, language areas, for example, seems to be highly variable. We do not yet the precise relationships, but this is a factor in which we are interested.